Good morning, students. In today's class, we'll move on to the last part of Transdermal drug delivery system, that is different evaluation parameters for TDDS. First part is a physical evaluation of TDDS. In that, first parameter is film thickness. Thickness of the film is measured by using an electronic vernier calipers. It is done with a least count of 0.01 micrometer. Thickness is measured at 5 different points on the film and an average of 5 readings is taken. Second evaluation parameter is percentage flatness. So here in this the film is cut into strips, 2 from either, 1 from center. Length of these strips is measured to the nearest centimeter without applying any additional pressure. The percent flatness of the strip is selected as the average percentage of length calculated from the 7 centimeter strips. Next parameter is patch thickness. Patch thickness can be measured using a digital micrometer screw gauge at 3 different places. Then the mean value is calculated. Next parameter is folding endurance. Folding endurance of the patches can be determined by repeatedly folding a small strips of film 2 into 2 centimeter at the same place till it breaks. The number of time the film could be folded at the same place without breaking is the folding endurance value. Next parameter is tensile strength. The tensile strength can be determined by using a modified pulley system. Weight is gradually increased so as to increase the pulling forces till the patches break. The force required to break the film is considered as a tensile strength. It is calculated as kg per centimeter square. So these are the first evaluation of TDDS. Coming to the second part of evaluation, that is weight variation. Weight variation is studied by weighing 10 randomly selected patches, and any variation in the weight can be calculated or evaluated. Next parameter is drug content. So here the patch is taken, 5 cm square film is cut into small pieces. It is put into a 100 ml buffer, that is pH 7.4, and shaken continuously for a period of 24 hours. Then whole solution is ultrasonicated for 15 minutes. After filtration, the drug is estimated spectrophotometrically and drug content is determined. Coming to the next parameter is a percentage moisture content. Here the films are weighed individually and kept in a desiccator containing an activated silica at a room temperature for 24 hours. Individual films are weighed repeatedly until they showed a constant weight, and a percentage of moisture content is calculated as a difference between the initial and the final weight with respect to a final weight. Next parameter is a percentage of moisture uptake. A weighed film kept in a desiccator at a room temperature for 24 hours is taken out, and it is exposed to 84% relative humidity, which is having a saturated solution of aluminum chloride. It is kept in a desiccator until a constant weight for a film is obtained. The percentage of moisture uptake is calculated as a difference between a final and an initial weight with respect to a initial weight. Next parameter or evaluation parameter is adhesions to steel, that is peel adhesion test. The measure of patch strength between an adhesive and the substrate is defined as a adhesion. Peel test is one of the standard tests used to evaluate the strength of adhesive patches. These properties are measured using 180-degree peel adhesion test method. Next is skin irritancy studies, erythema. This study can be conducted on mice or albino rats. The potential of transdermal system can be evaluated by modified Drey's test. Here the scores are given from 0 to 4 depending on the degree of erythema as follows. If there is no erythema, then the score is 0. If it is slight, that is barely perceptible light pink color, it's considered as 1. For a moderate, it should be dark pink, then it is scored 2. For a moderate to severe, then it will be light red, it is scored 3. If it is severe, then it is extreme redness, it is scored 4. Next evaluation parameter is confocal laser scanning microscopy. Here the depth of the skin penetration of a patch can be assessed using this test. Transdermal formulation is applied non-occlusively for 8 hours to a dorsal skin. The mice is sacrificed by heart puncture. Dorsal skin is excised and washed with distilled water. Then the excised skin is placed on aluminum foil and the dermal side of the skin is gently teased off any adhering fat and subcutaneous tissue. Then these are cut into pieces of 1 mm square and tested for probe penetration. The full skin thickness is optically scanned at different increments through z-axis of a microscope. This was about a laser scanning microscopy. Coming to the next one, that is skin permeation study. The in vitro skin permeation of transdermal system can be studied using a France diffusion cell. It is done with an effective permeation area and a receptor cell volume of 1 cm square and 10 ml respectively. Temperature is maintained at 32 degrees Celsius. The receptor compartment is filled with a 10 ml solution and is constantly stirred in a magnetic stirrer at 100 rpm. Skin is then carefully checked through a magnifying glass to ensure that samples are free from any surface irregularities like tiny holes or cervices. The skin is mounted on a receptor compartment with the stratum corneum side facing upward into a donor compartment. Transdermal system is applied on the skin in a donor compartment and the samples are withdrawn through a sampling port of a diffusion cell at a predetermined time interval over 24 hours and are analyzed. The receptor phase is immediately replenished with the equal volume of fresh diffusion buffer. So this is a schematic diagram of a France diffusion cell where you can see dosage donor area, glass disc, membrane is placed, water is placed, receptor solution, sampling port you can see, water jet which is maintained at temperature 32 degrees Celsius, stirrer is present and you have to replace a port with a bubble trap. So this is a schematic representation of a France diffusion cell. Coming to the conclusion, improvement in transdermal drug delivery system is a successful story of a pharmaceutical endeavors. Several transdermal patches are already in market, which is valid proof that such systems are feasible, safe and effective with a maximum patient compliance. Microneedles will be further improves the application of transdermal drug delivery system. Ontophoresis and sonophoresis may help widening the spectrum of the drug candidate for such drug delivery system. So this completes the chapter of transdermal drug delivery system. Thank you.